Good morning, bio students. This is Praful Sharma, biology lecturer, TUP School Amed. Let's start our today's session for chapter number 2, male and female gametophytes, structure and development. As we have already talked about that flowers are formed as reproductive organ in angiosperms, and morphologically flower is a modified shoot with limited growth in which the flower whorls and nodes are highly condensed. Therefore, the flower is also called a sporophyll-bearing shoot. In our today's chapter, we will talk about the two essential whorls of flower, that is androecium and gynoecium, their structure, their development in more detail. Let us start with the structure and development of male gametophyte, stamen. Stamen, which is also known as microsporophyll. Each stamen is divided into two parts. You can see this thing simply in the diagram which is shown here, that a stamen is divided into two parts. The upper part is the lobed structure. We call this structure as anther or pollen sac, and the down long stalk through which this pollen sac is attached is called filament. The pollen sac we are visualizing here is bi-lobed. In Malvaceae family, there is a characteristic feature with that family is that they have a mono-lobed, single lobe of pollen sac is there, but in most of the angiospermic flowers, it is bi-lobed. This bi-lobed pollen sac has two lobes, and these two lobes are attached with each other through a connective tissue. This connective tissue is named as connective. This connective is completely sterile part. All parts of this microsporophyll are reproductive in nature. Each lobe of this pollen sac is further divided into two chambers. These chambers are called pollen sacs, or they are also known as microsporangia. Look at this picture and see that each bi-lobed pollen sac contains 4 microsporangia. The pollen sac in Malvaceae family is single lobed and it has 2 microsporangia obviously, and pollen grains or microspores develop in microsporangium. The diagram here shows that each pollen sac is further divided into 2 chambers. So a total of 4 chambered structure a microsporangium is. As you all know that every living body is made up of cells. A plant is also a living organism. It is also made up of cells. In initial stages, when the flower is in a bud stage, it's just a homogeneous mass of multiple cells. All these cells are similar or alike in nature. When they are in maturation period, the internal central cells, mass of cell, becomes more denser in cytoplasm, and these specialized cells are named as archisporial cells. The archisporial cells shows periclinical division, means these cells they divides in such a way that outer side they develops primary parietal cells and inner side they forms primary sporogenous cells. The archisporial cells, when they divides and at the outer side they forms primary parietal cell, these all primary parietal cell forms the pollen sac wall, while the primary sporogenous cell which are formed inside forms the microspore mother cell, or divides mitotically to produce many sporogenous cells which later form microspore mother cells. Thus, a pollen sac divides into 2 parts after developments: pollen sac wall and sporogenous cells. Let us talk about the pollen sac wall. This wall obviously is formed or designed by the primary parietal cells which are formed from the periclinal division of archisporial cells. The pollen sac wall is formed from primary parietal cell which are formed at the outer side by archisporial cell division, which later on at the maturity shows 4 layers in a cross section. These layers are arranged from periphery to the center in a definite sequence as follows. The outermost layer is a single layer of outer cell which provides protection to pollen sac. These cells are epidermal cells. Epidermal cells of mature pollen sac elongated and flat in the direction of contact line, and this is the first layer of this pollen sac wall, and it is termed as epidermis. The second layer inner to epidermis is endothecium. It is a single layer of cells below the epidermis. These cells are thick and radially elongated. In the internal wall layers of the cells, at the contact line, alpha cellulose gets deposited, due to which fibrous strips are formed. The third layer of the pollen sac wall is middle layer. They are 1 to 3 numbers. Their layers are 1 to 3 inside the endothecium, and they are actually short-lived and thin-walled. The last and the fourth layer of pollen sac wall is termed as tapetum. It is the innermost single and special layer of cells that surrounds the sporogenous tissue. This layer completely develops by the time sporogenous cells reach tetrad stage. We'll talk later on about this. The cytoplasm of tapetum cell is dense and nucleated. During the maturity time, these cells become multinucleated and polyploid. In angiospermic plants, on the basis of behavior of cells, tapetum is of two types, which are known as amoeboid or periplasmodial tapetum and secretory or glandular tapetum. Tapetum is quite different. It has multiple features to do and perform. Let us talk about the next one, the sporogenous cells. They are also formed by the periclinical division of archisporal cells, and they are forming inside at the center position of this whole structure, which we call as anther. The group of cells covered with another sac wall is another sac are called sporogenous cells. These cells produces microspore mother cells. By meiosis, each spore mother cell produces 4 microspores. So in this way, we just understand that when a flower is in bud position, it has multiple type of cells, but all these cells are quite alike, homogeneous mass of cell. Out of all those cells, the centrally aligned cells becomes specific. They have more denser cytoplasm, and we started calling them microarchisporal cells. These archisporal cells, when they start maturing themselves, they divide periclinically, and at the outer side they form primary parietal cells, and inner side they form primary sporogenous cells. The primary parietal cell in future will give rise to the pollen sac wall. This whole pollen sac wall is formed by these primary parietal cells, and the wall contains 4 layers, and they are epidermis, endothecium, middle layer and tapetum. And the second type of cell formed from archisporal cell, primary sporogenous cell, it divides and forms sporogenous cells. These sporogenous cells will become mature, divide by meiosis, and will give rise to Microspore Mother Cell, which in future will give rise to microspore, which will we talk about in our next session. Here we are done with our today's session. Guys, now it's your time to learn these concepts clearly and do this homework undersigned. Have a nice day.